We left off speaking about the general anatomy of the nervous system. We spoke a lot about the action potentials and a lot of the details about neurons and how they communicate. So we've really been zooming out and looking at what the anatomy of the nervous system looks like. And this is a lot of overlap from things we would have talked about in lab, okay? So let's pick up here with the blood supply to the CNS. And as we talk about this, you really should be connecting the circle of Willis. We talked about the circle of Willis in lab, and that really reflects the needs of the nervous system, the needs of the brain. The brain is highly metabolically active, okay? Although it accounts for a very small amount of our body weight, about 2%, okay? So brain and spinal cord together are not very heavy, not very much in terms of their weight and their percent of our body weight, but they take up quite a bit of our metabolic activity. Okay, and so even at rest, the brain uses about 20% of the oxygen that we breathe in and 50% of the glucose that we take into our diet. So the brain is very, very demanding in that way in terms of its blood flow, oxygenation, and glucose supply. And this is partly because the brain cannot use glucose in a way that other tissues can. It cannot store glucose, right? There's very little glucose or glycogen reserves. And then glucose cannot easily move across the membrane. We'll talk about the blood-brain barrier and why that happens. Okay, so these are some of the needs of the brain: very heavy oxygen demand, very heavy glucose demand. Let's talk about what happens in terms of the blood metabolism in the brain. So the neurons in the brain rely on aerobic glycolysis, meaning that they must break down glucose in the presence of oxygen, okay? They cannot undergo that oxidative phosphorylation the way our other tissues can, muscles and so on. So it requires a steady supply of glucose and oxygen for this reason. And it doesn't store glycogen very well, again, the way our muscles can, liver can, other tissues can tap into those glycogen reserves. The brain cannot do that. So it must have a continuous flow of glucose coming in. It can use ketones, which are the breakdown products of fats. It can, but inefficiently and only in extreme circumstances can the brain switch over to this. And it's not very effective. It won't last very long. Eventually, you will result in coma and death with no oxygen and no glucose flowing to the brain. When we have lack of blood flow to the brain, this is called a stroke. Okay, so this can be for two different reasons: either an interruption in the blood supply, such as a blockage somewhere. That typically happens when there's plaque in the vessel, or if there's some sort of hemorrhage happening, right? There's a rupture to the vessel, which typically is seen with high blood pressure and those types of things. Okay, so different types of strokes, but whenever we lose blood supply to the brain tissue, this is called a stroke. Let's talk about the anatomy of the blood-brain barrier, okay? Now, what is the blood-brain barrier? It's the site of exchange between our central nervous system and our general circulatory system. And so this is basically limiting that exchange, okay? It's a single layer of endothelial cells, and endothelial cells are the cells that line our blood vessels. They are epithelial cells, specifically around blood vessels. We call them endothelial cells. And they facilitate some diffusion of molecules, but they're very selective in what can get through into the brain, okay, and the nervous tissue. And so it's formed by these astrocytes. We talked about these astrocytes, these star-shaped cells, and I told you to sort of imagine them as being a gatekeeper for the nervous system, and that's their function, and that's the way they look as well. So this is your blood vessel. This is a blood vessel that's in the brain, and sitting around that blood vessel are these astrocytes. And these astrocytes have these foot processes, right, that we call podocytes, these little horizontal processes that actually prevent certain materials from leaving the circulatory system, leaving the vessels in the blood and making their way into the CSF, into the internal or the interstitial environment in the brain, okay? So the blood-brain barrier is formed by these two structures, the tight junctions that are naturally formed between endothelial cells. We spoke about tight junctions at the beginning of the semester. They're at the lateral domain of two cells, and they prevent leakage or diffusion of materials. So the tight junctions, along with the foot processes of the astrocytes, together forms that very selective barrier. Okay, this is really important. This is evidence of how sensitive the brain is, that it has to select or barrier itself from the rest of the body. This prevents unnecessary bacteria, pathogens. It even prevents unuseful substances. So hormones, neurotransmitters that don't need to be in the brain should not be there, okay, and they're kept out. In terms of what can get through here, we know that some hydrophilic solutes, very selective, can get through, and then some hydrophobic solutes that can freely move through this membrane can get through as well. Now, the hydrophilic solutes, as we know, need to be transported or carried, right? They can't just go through that membrane. And so there are some pumps and carriers along the membrane here to facilitate that. But what we know that doesn't get out are glucose to some extent and proteins, right? Proteins are too large to get through this junction, to get through this barrier, and this is why we don't see them in the CSF. We talked about those relative proportions of protein and glucose in the CSF, right? The CSF is deficient of protein, deficient of glucose, and this is part of the reason why. Okay, so the important takeaway point here, astrocytes, right, standing as the gatekeeper, protecting the nervous tissue, and the the tight junctions between the endothelial cells helping that process as well. All right, the last thing we're going to speak about is myelination. Myelin, we've spoken about the function of myelin as it pertains to propagating the action potential, and here we'll really talk about how that myelin comes to be. Now, in the central nervous system, myelin is formed by oligodendrocytes. Those were one of our 5 types of glial cells that we introduced last time. Okay, oligodendrocytes. And these specifically myelinate the central nervous system, i.e. brain and spinal cord. Okay, and the way they do that is that they produce this myelin sheath. Okay, and I'm going to say right here early, this is not a neurolemma. And I'll explain in a minute what differs this from a neurolemma. Okay. So oligodendrocytes sit in between several neurons, and the cell body's not interacting with the neuron itself, but they're sending off these axonic processes that engulf the neuron and then myelinate it, okay? So the axon processes of the oligodendrocytes is what is actually myelinating the neurons. We know that myelin gives tissue this white appearance, right? Especially when we stain it. Myelin is mostly phospholipids and fat stains kind of white. And so this is called white matter. Whereas unmyelinated tissue lacks this myelin, and so it stains a bit darker, and we call it gray matter. And so if you look at a cut section of the brain, we can distinguish this gray matter, which sort of coats the overtop of the brain, and the white matter, which is closer to the inside. Now, in terms of the actual part of the neuron that's in white matter and gray matter, we know that the axons themselves are the white matter, that's the projecting part, whereas the cell bodies, the soma, the dendrites are going to be the white matter, right? We know that there's no myelin around this part of the neuron, so that's going to be your gray matter. Your gray matter, right? So I'll say that again. Axons are going to be white matter because they're myelinated. Dendrites, soma, which is the cell body, is not myelinated, and so we call it gray matter, okay? Okay, now when you look in the peripheral nervous system, this is different. Okay, we don't have oligodendrocytes. What we have are Schwann cells, and we do create a myelin sheath, but we also have a neurolemma, which is a little bit different from the myelin sheath. So Schwann cells create this neurolemma, which is basically how we insulate these peripheral nerves. Now bear in mind, this is outside of brain, outside of spinal cord. Okay, so what we have is a Schwann cell. This is its nucleus here. This is its plasma membrane, which is essentially the neurolemma. So we can think of the neurolemma as the membrane of the Schwann cell. Now here, the Schwann cell is doing a different thing than the oligodendrocyte. It's actually hugging the neuron. So the entire cell is wrapping itself around the neuron, as opposed to oligodendrocytes, which send off axonic processes that coat the neuron, okay? And so what happens here is now this neurolemma, which is the membrane of the Schwann cell, starts to wrap around this plasma membrane. So we can see it's not very well developed here, but by the time this is sitting around this neuron for a while, we develop this wrapping of the plasma membrane, and that is the myelin. Okay, so 2 important distinctions to make here. One is that the myelin sheath is different from the neurolemma. The neurolemma is basically the membrane of the Schwann cell. Okay. 2 is that the myelin here is the actual plasma membrane that's wrapping around the axon. Okay. So in the middle here, this blue portion is the axon, and as this Schwann cell sits around it, wraps that neurolemma around it, we start developing that wrapping of the plasma membrane around the axon, and that's how we myelinate in the peripheral nervous system. Now we know that the gaps between this myelination are called nodes of Ranvier, and this facilitates that saltatory conduction where action potentials leap from node to node. We talked about this earlier. So at this location, there are excess voltage gated sodium channels, and this facilitates the leaping or the jumping of the action potential from one node to the other node to the other node in that saltatory fashion. Okay, we spoke about that. In terms of looking at unmyelinated axons, they still have a neurolemma. Okay, and this is where distinguishing the neurolemma from the myelin becomes very important, because although they don't have myelin, they still have a neurolemma. They have that Schwann cell wrapping around them, and remember we said the neurolemma is the plasma membrane of the Schwann cell. But they haven't myelinated, they haven't wrapped that plasma membrane around the axon, and so although they have a neurolemma, they don't have myelin. Okay, this darker staining region here is showing what a myelinated axon would look like. Okay, now we know the difference here in terms of function is that myelinated axons conduct much more rapidly. They have a faster conduction velocity, and so in smaller axons like autonomic neurons, neurons that innervate the heart, our glands, our GI system, they're not myelinated and they're much smaller in their diameter, okay? And so they're gonna conduct faster. Peripheral nerves that innervate skeletal muscle are gonna have a much thicker diameter, are gonna be much more well myelinated and to produce or propagate that action potential faster.